Hello, everyone. Today we are starting a very high yield topic, gastric secretions. This is not just physiology or biochemistry, it's a clinical backbone topic. If you understand this lecture properly, you will be able to handle peptic ulcer disease, acid related disorders, drug mechanisms like proton pump inhibitors, and H2 blockers. So stay focused. This is the core material, right? What about the learning objectives of this session? By the end of this session, you should be able to describe the gastric juice contains, what gastric juice contains, explain how each component functions, understand what stimulates and inhibits gastric secretion, and most importantly, the mechanism of HCl secretion, right? If you want to think clinically, or I want you to think clinically, whenever acid changes, something pathological is happening. What about the learning resources? These concepts are aligned with Lippincott for conceptual clarity, Chatterjee for deeper biological details. But today, I will integrate everything into a clinically usable framework. Let's start with its structure. The inner surface of the stomach is not smooth. It holds folds like rug-like structures. Within these folds, we have gastric pits. These pits often or open into gastric glands, which are the real functional units. Think of this as surface of the surface as an entry and gland as factories. Let's discuss the lining of the gastric pits. Now, this is extremely important because each cell has a specific function. Very first one is surface alveolar cells. These cells secrete mucus and bicarbonate. Their job is protection. Without them, acid will digest the stomach itself. Next is mucus neck cells. They produce mucus but also act as stem cells. They also or they maintain the epithelial lining. Next is chief cells. They secrete pepsinogens. Pepsinogen is inactive. It needs HCl to become pepsin. Next is the parietal cells. Now focus here. This is the star of today's session. Parietal cells secrete hydrochloric acid, HCl, and intrinsic factor. And intrinsic factor is essential for vitamin B12 absorption. If the parietal cells are damaged, pernicious anemia would be there, would occur. Next is the G cells. These are endocrine cells in the antrum. They secrete gastrin, which stimulates acid secretion. So this is hormonal regulation. Every day, stomach secretes about 2 to 3 liters of gastric juice. The pH is around 1 to 2. This is extremely acidic, comparable to battery acid. So, functionally, this acid denatures proteins and kills bacteria. Let's discuss the composition of gastric juice. Gastric juice is almost or mostly water, about 99%. The remaining include inorganic and organic components, right? Inorganic components may include sodium, potassium, hydrogen, magnesium, chloride, bicarbonates, phosphates, and sulfates. And organic components or constituents may include mucins, pepsinogen, gastric lipase, intrinsic factors, right? And mucus as well. If you see lactic acid increasing, it suggests low acid and bacterial growth. Let's discuss the functions of HCl. Let's break this down clinically. Very first one is activation of pepsin. HCl converts pepsinogen to pepsin. Second, it provides acidic pH, optimal environment for protein digestion. The third one is antibacterial role. It kills ingested microorganisms. Number 4, iron absorption. It converts ferric Fe+3 to ferrous ions Fe+2, which is absorbable form of iron. Number 5, hormonal role. When acid enters duodenum, secretin releases, and secretin enhances the pepsinogen secretion. However, the increased level of secretin inhibits the acid secretion. Let's discuss the heart of this lecture or this session. If you understand this, you are exam ready. This is the mechanism of HCl secretion. This is the core of this session. Let's discuss step by step. I would divide into multiple steps, right? Step number 1. Step number 1, inside parietal cells. Carbon dioxide diffuses into the cell and reacts with water. This forms carbonic acid, H2CO3. Step number 2, carbonic anhydrase action. This enzyme, carbonic anhydrase, is extremely important. It accelerates the reaction. Step number 3, dissociation. Carbonic acid dissociates or splits into hydrogen ion and bicarbonate ion, H plus and HCO3 minus. Step number 4, this pump or the proton pump actively secretes the hydrogen ion into the lumen. This is the final step of acid secretion, and this is where proton pump inhibitors, the medicine or the drugs, act in order to control the acid secretions. Step number 5, chloride movement. Bicarbs, those are produced by the dissociation of carbonic acid. Bicarbonates or bicarbonate ions leaves the cell into the blood, and chloride enters. Then chloride diffuses into the lumen. And step number 6, the formation of HCl. In the lumen, hydrogen ion and chloride combines to form HCl. Okay, now this is the alkaline tide or clinical concept related to mechanism of HCl secretion. After meals or every meal you take, bicarbonate enters the blood blood becomes slightly alkaline. This is called alkaline tide. This is the illustration of the mechanism of HCl secretion. As you can see in this illustration, carbon dioxide. Carbon dioxide diffuses into the cell, it reacts with water by the action of carbonic anhydrase, Ca. It splits into hydrogen and bicarbonate ions. Bicarbonate leaves into the cell and chloride enters, right? And in the lumen, H and Cl ions combine to form HCl. And after every meal, bicarbonate enters the blood and blood becomes slightly alkaline. This is alkaline tide. Thank you very much. If you found this session useful, revise the mechanism once again and try to draw it yourself. That's the key to retention. In next lecture, we will discuss further on gastric biochemistry or gastric content on the stimulation or stimulants and depressants, along with the clinical relevant conditions of gastric secretions. Thank you very much.